All right, how's it going, everyone? My name's Andy Feroriouk. I'm a PhD student starting in the Shikuma lab at San Diego State University. I'm going to be talking about my prospective PhD project. So we study Hydroides elegans with the objective of making genetic tools for this new model organism. So colloquially, Hydroides elegans is known as the tube worm. So here's a basic life cycle of Hydroides elegans. It's representative of other marine organisms. So you got the eggs and the sperm. These guys combine and they make the larva over here. This is our larva. Okay. So the Hydroides elegans larva is interesting because it undergoes a dramatic life history transformation, so-called metamorphosis. I'm thinking of the Franz Kafka book. Someone got it. All right, so the metamorphosis from the larva into the adult stage over here. So the adult stage settles in a benthic marine environment, and it's a filter feeder organism, okay? So what's most interesting about metamorphosis is, first of all, how dramatic it is, and second of all, how complex it is. So a lot is not known about metamorphosis, but what is known is that the process does require bacteria. So you have marine bacteria over here, and really there are many bacteria can induce metamorphosis, but the one of particular interest to the Shikuma lab and the one which Dr. Shikuma has pioneered numerous genetic tools for already is called Pseudo-Ultramonas luteoviolacea. Pseudo. Okay, so colloquially you call it just P. luteo. So this bacteria over here, it produces structures, protein structures upon lysis, look like these spiky balls, and these are actually syringe-like structures which have some homology to phage structures, and these guys inject themselves into the Hydroides elegans larva. Okay. All right. And they release a payload, which then causes cell signaling, okay, inside the Hydroides elegans, and that facilitates the metamorphosis. Okay. So our objective in this project is to use microinjection over here, or electroporation, all right, to insert transgenes, plasmids, okay, plasmids into the Hydroides elegans via either gain of function or loss of function approach. Okay. All right. And our first question is, okay, how does this impact the metamorphosis capabilities of the Hydroides elegans? And the second question is, okay, what is or are the changes in the cell signaling that occur in the Hydroides elegans larva when you introduce these transgenes, and how does that impact the process? So the novelty of this project rests in adapting a novel model organism, the Hydroides elegans, with two biological and molecular biological techniques, and leveraging Dr. Shikuma's previous experience with the bacterial genetics in order to investigate this process completely and ideally learn something more about symbiosis between different kingdoms of life, and then how we can far in the future leverage this for some kind of therapeutic benefit, or for instance to benefit fuel economy in ships, which are often crowded with these guys, and that causes, you know, dramatic loss of fuel, things like that. All right, so hopefully we'll develop some genetic tools. Thanks for your time.